Today we're going to be discussing DNA replication, which occurs in the nucleus during interphase. DNA is made up of two strands in a double-stranded helix formation. It runs 5' to 3' and they are complementary. For DNA replication, there's an enzyme called helicase which unzips the strands. DNA polymerase builds DNA molecules using RNA. Primase makes the primer from RNA for polymerase to work, and ligase glues fragments, the Okazaki fragments, together at the end. Telomeres are put at the ends of the strands and they protect our DNA. They're responsible for aging and also cancer, and they are important in filtering through garbage DNA. So for replication, it starts at the origin of the DNA, and it starts with helicase unwinding the DNA into two strands, a leading strand and a lagging strand. And then primase creates the primer for polymerase. Polymerase 1 makes DNA. Polymerase 3 sits on the primer and works until it runs into an Okazaki fragment. Polymerase 1 then replaces and removes the primer. And lastly, ligase connects the Okazaki fragments.
Okay, I'm going to be going more into detail on the steps of DNA replication. So the first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA, and an enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer, and this marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of the DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as a semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one.